Let's say that Tommy and his dog Buster are taking a little walk. Suddenly, Tommy has a question. Are he and Buster really that different? Is there something fundamentally the same between him and his buddy, besides the fact that they both love peanut butter? Is there a scientific way we can address Tommy's question? The answer is yes. Inside Tommy and Buster's cells, tiny little particles contain the building manual for their physical makeup. These tiny little particles are called DNA and are composed of two strands of letters zipped together. The order of the letters form a kind of instruction book that gives Buster his black nose and Tommy his red hair. If an identical strand of letters are found in both Tommy and Buster, it means that they have something fundamentally in common. Let's say that inside Tommy's muscle cell he has a piece of DNA known as piece Y from a muscle protein gene. How do we know if Tommy and Buster have similar or different piece Ys? To see, we use a method called southern blotting. In order to start identifying piece Y, we cut Tommy and Buster's DNA into smaller fragments. Piece Y is somewhere in here. In order to envision piece Y more clearly, we must separate it from all the other fragments that we also cut. The best way to do this is to separate the different fragments by size. Smaller fragments move faster than larger fragments when pulled by the same force. If we imagine Tommy and Buster racing, with Tommy carrying a magazine and Buster carrying 5 textbooks, Tommy will show up at the finish line first. We separate the DNA fragments similarly by racing them on a gel with tiny spaces, with negative DNA being pulled by a positive force. Larger DNA is heavier and will move more slowly than smaller DNA. The gel at the end will look something like this. Tommy's lane is on the left and Buster's on the right. We then transfer the gel onto a sponge and put it in an alkaline solution. A nitrocellulose paper is put on top of the gel and paper towels to weigh the whole thing down. The solution is drawn up from the sponge and effectively unzips our DNA fragments into single strands. They are then absorbed onto our nitrocellulose paper. So we have a paper full of single-stranded DNA, and our fragment Y is somewhere on there. Both Tommy and Buster's DNA fragments are separated and shown here, but how do we know which one is fragment Y and if Buster has the same fragment Y as Tommy? Well, remember that DNA is composed of two strands zipped together. Unzipped DNAs will look for its other half to pair back together. Relying on this fact, we make a single-stranded probe identical to Tommy's fragment Y. Our probes will only reconnect to their complementary strand. We sprinkle our tagged probes onto the paper full of unzipped DNAs and go join Tommy and Buster for a little picnic. After we have come back and developed the paper on an X-ray film, we see the work that our probes have done. There are 2 dark bands on the paper at the same relative spot in both Tommy and Buster's lane. That means that our tagged piece Y really rezipped with Tommy's fragment Y, as expected, but also Buster's as well. That means that Buster has a fragment Y that's the same as Tommy's. Finally, they have something in common that they can talk about.